Hello everyone, I hope all of you are doing fine. Today we will be giving a webinar about a very important topic, how Fapon's raw materials are leading to high performance respiratory antigen test. I am Sandeep, I'll be giving you this webinar. I am the Senior Global IVD Product Director at Fapon Biotech. One of my colleagues, Will, helped a lot in drafting this presentation for the webinar. Now let's discuss first about Fapon. As most of you might already know, Fapon is one of the topmost IVD raw material and IVD solutions provider. We have been in existence for more than 23 years. The company originated in 2001 and we started producing antigen, antibodies, and the enzymes for the entire IVD industry. Later on, we transitioned into the IVD applications, where apart from the raw materials, we also started looking into the reagent solutions. Further on, from 2017 onwards, we started the one-stop solutions for the IVD industry, where apart from the raw materials and the reagents, we also started providing the open instruments to the IVD manufacturers. And from the last 4 years, from 2021 onwards, we are directly working in collaboration with many of the IVD manufacturers, where we are targeting innovative IVD applications together with our industrial partners. And for the future, we will continue our journey for innovation and exploration and targeting new IVD spaces. At this moment, Fapon has more than 1100 patents that encompass most of our IVD technologies as well as the product solutions that we are providing to the industry. We have more than 1,400 matured IVD products, and globally we have more than 2,500 industrial partners. And we are targeting multifarious assay formats, whether it is the fully automated chemiluminescent immunoassay, ELISA, fluorescent immunoassay, colloidal gold, rapid test, NGS, LAMP. Now, in this particular presentation, we will be discussing about the respiratory infections and how they are detected. Then we will be discussing about the development of the influenza antibodies at Fapon. Subsequently, we will discuss about the raw materials for the respiratory antigen test that Fapon has developed and providing to the IVD industry. And lastly, we will discuss about how we are prepared for the future. So first, let's discuss about the respiratory infections and how they are detected. Most of you already know that respiratory infections are a big global threat. And we have seen like the COVID-19 pandemic, during which millions of persons have lost their lives. And lower respiratory infections, they account for one of the leading causes of that worldwide. So it is the fifth leading cause as per the WHO report. And it is one of the most deadly communicable disease. In the next slide, we will be discussing about the clinical detection of the respiratory infections. This one is taking an example from COVID-19. Before the onset of the symptoms, that is like the incubation period, it is very difficult to detect the respiratory infections. And most of the subjects are asymptomatic. So the detection of the respiratory infections is very unlikely during this particular stage. But after the onset of symptoms, there is a very high likelihood for the detection of the respiratory infections. You can detect the virus via the RT-PCR using the different kinds of the swab. Most commonly used is the nasopharyngeal swab. And you can also detect the virus via the respiratory antigen test. But starting from 2 weeks post onset of the symptoms to up to 4 weeks, there is a very high likelihood for the antibody response also. So you get your IgG, IgM as well as the IgA response. And during this particular time period, you can detect most of the respiratory infections. So from the start of the onset of the symptoms to around 4 weeks post onset of the symptoms. You can detect most of the respiratory infections using the RT-PCR test, respiratory antigen test, and you can also get a good idea about the antibody response of the subjects. And after that, like, the subjects goes into the convalescence. So IgG stays like for a long period of the time, but IgA and IgM response started decreasing. Now in the next slide, let's discuss how the various respiratory infections are detected. So there are two types of tests. One is like the antigen test, and the two common type of the antigen test are the lateral flow assays and the direct fluorescence assays. Most of the antigen test, as you already saw during the SARS-CoV-2, they are used for the population screening. The assays are very cheap, easy to perform. You don't require any instrument for the readout, and everyone can understand the test results. Similarly, there are other tests which are based on the RT-PCR. So the molecular diagnostics are the confirmatory diagnostics, but they are highly expensive. Nearly, you can say 40 to 50 times more expensive than the RT-PCR, and they consume more time in comparison to the respiratory antigen test that takes a couple of minutes. The molecular diagnostic test in the central lab that can take like a few hours, and the point of care molecular diagnostics that is nearly 100 times more expensive than the respiratory antigen test. But they can take you time span of less than 1 hour. So depending on the applications where you want to apply, whether it is for the population screening, whether it is for confirmatory diagnostics, whether it is developed countries, developing countries, or you need to test very fast, or you have specific time window and more flexibility in terms of the resources. So you can detect the respiratory infections via these two means. Now, in the next slide, we will be discussing about the global respiratory infections. The international community is already aware these respiratory infections are prevailing on a large scale worldwide. COVID, all of us, we already know. Influenza A and B, we already know from many years. This is one of the most common respiratory infections worldwide. Then we also know about the RSV, ADV, as well as MP. So these are the most common type of the infections, respiratory infections prevailing worldwide. There is a need for point of care applications for the rapid detection of these particular infections, and especially when it is a pandemic or the epidemic scenario. The point of care diagnostics is very much relevant because you can screen a bigger population and very, very fast without any need of any instrumentation that is costly and expensive, and it is very easy to understand. But still, point of care tests have some significant challenges in terms of the sensitivity, in terms of the cross reactivity, and to go from screening to the confirmatory, there is a lot of testing still required so that it can match the accuracy and the sensitivity required for the confirmatory diagnostics. But a lot of improvement has happened in the past years, and I think like most of the point of care tests in the near future are progressing rapidly towards the confirmatory diagnostics. And we will soon see many of the technology improvements and how IVD manufacturers are targeting these particular limitations also. Now, some of the recent trends that have emerged during the past years. At the start of the COVID pandemic, most of the rapid tests for the respiratory infections, mainly for SARS-CoV-2, they were given the emergency use authorization. So you can see hundreds of tests that were given the emergency use authorization. Now from May 2023, FDA has formally terminated the emergency use authorization, and most of the IVD manufacturers that had the EUA, they have to transition to the regular route of the FDA, that is the 510K, or if they fall under the de novo or the breakthrough denomination, they can go for those particular routes. One of the particular developments was from Helgen, that was given the de novo clearance by FDA for their COVID-19 influenza A and the influenza B triple test. Most of the IVD manufacturers that are developing like the multiplex test for COVID-19, influenza A or influenza B, they now have to evaluate their kit with respect to the kit from Helgen, keeping it as a predicate. Fapon played a big role during the COVID time, and most of our raw materials have been used by the topmost companies for the development of the IVD solutions for the respiratory infections. From our experience during the pandemic and from the way we are developing the raw materials for the respiratory infections, we can help the IVD manufacturers a lot and help you in developing the market-ready assays for the FDA or IVDR by decreasing your time to the market. Now let's discuss about the development of the influenza antibodies at Fapon. As most of you already know, that designing the influenza immunogen is very, very tricky. There are too many strains of influenza virus that are already prevailing. When we develop the immunogen for the influenza virus, one of our core objectives is it should be capable of detecting all the multifarious strains of influenza that are already prevailing. We study about the conservative sequences for the influenza strain and select the most prominent sequence containing all the prospective epitopes that are required for the successful detection of the influenza virus. In case of influenza, our target protein is nucleoprotein, and we see the nucleoprotein sequences among all the influenza strains and select the most conserved sequence containing the epitopes for the detection of the influenza. In the end, our immunogen is the recombinant nucleoprotein derived from the H3N2 subtype of influenza. In this particular slide, we will discuss about the production of the influenza immunogen. In the first phase, we do the protein design and employ the genetic engineering technology. Thereafter, we select the appropriate expression system, that is E. coli, yeast, or the CHO cells. In the third stage, we go for the purification and the maturation. And finally, we go for the detection and the screening, employing various technologies. In the next slide, we discuss about the core technology and the process, how we develop the influenza antibodies. We start with the immunization of the animals. Then we have different technologies. We have the conventional hybridoma, and we have the multiple generations of the single B cell technology. And presently, Fapon is employing the single B cell technology with artificial intelligence that enables us the development of the antibody very, very fast. We can determine the antibody binders just in less than 1 week. So once we have done the discovery of the antibody binders, then we go for the expression of the antibody. And we find a most suitable cell line for the development of the antibody. Then we go for the large scale fermentation in our bioreactors. Finally, we go for the purification of the antibodies and we analyze what we have produced using the various physicochemical analysis tools at our disposal. We check in the final stage whether the antibody target specifications we have successfully achieved. Next slide is about the preclinical testing. So the influenza antibodies that we have developed with the particular technical specifications, we want to check whether they can detect all the circulating influenza strains. We check them using a wide variety of the PCR validated samples, clinical samples that we have at our disposal. We check it using the various quality controls, using the recombinant antigens and using the different titers. Then we employ like checkerboard screening, during which we identify 3 to 5 best antibody pairs that we have identified during the development, and we check them how they are holding up in terms of the rigorous testing parameters that we are doing here. We are also doing a rigorous cross-reactivity analysis against flu A, flu B, COVID-19, wide range of the drugs that can interfere with our target analyte, and also several other pathogens that can interfere. So we have in-house a comprehensive screening and testing for around 114 different strains of the influenza. So finally, whatever passes our preclinical testing, we already ensured that the IVD manufacturers will get the expected results for their respective IVD test, because the antibody pairs that we are developing, they are free of any cross-reactivity and they are highly sensitive for the respective target analyte. Then in the next phase, we will discuss about the raw materials for the respiratory antigen test. Now, first we will discuss about the influenza antibody performance evaluation. When we evaluate our antibodies, the first thing we see is like they have the desired analytical sensitivity. In our case, the antibody pairs that we have developed, they have very high analytical sensitivity. So you can detect 114 different strains of influenza. We have checked it on the NIPSC panel as well as the Zeptometrics panel, and all these strains and the panels we have successfully detected at high analytical sensitivity. And we are employing a wide range of the PCR validated clinical samples. So our developed raw materials for the respiratory antigen test, they have high clinical sensitivity. You can see like the PPA and the NPA for flu A and the flu B, where we have high clinical sensitivity. Further, we check our assays against the well-known rapid antigen test available in the market, and the performance from our raw material based test is very high. So they are matching the performance of the predicate kit and in some cases also exceeding them. That is one of the reasons that our influenza raw materials are used globally by the leading IVD manufacturers. And we have many of the commercial successes from the leading IVD players in the market. Now in the next slide, we will discuss about the performance of our COVID and the influenza raw material based test. We have achieved, as well as most of our IVD manufacturers, they have achieved high clinical sensitivity. So we have high PPA and the NPA, as you can see on the left side. We don't have any cross-reactivity. And we have checked all the interfering substances, pharmacological, as well as the pathogens that are closely related. We don't find any cross-reactivity issue by the use of our raw materials for the IVD test. And more than 30 IVD manufacturers, they have used our COVID and the flu antibodies, and they have successfully developed the COVID and the flu antigen test. And most of them, they have already achieved like the FDA as well as the CE approvals for their respective IVD test. So the performance of the COVID and the flu raw materials and the test based on them are really high. In the next section, we will discuss about our future preparedness. As all of you know, influenza is progressing very fast. So there is a need for real-time monitoring of all the influenza strains circulating globally. At Fapon, we have a team of scientists that is continuously monitoring the influenza activity, and we are checking every day, in fact, and going for an analysis every week, whether it could have an impact on the raw materials and the test that Fapon is developing. As soon as a new strain is reported, we will check the new strain using our in-house bank of the antibodies. If the new strain is detected successfully by our in-house raw materials, there is no need for us to go for the additional work. But if the new strain is not detected by the bank of antibodies that we have, we will design the new recombinant antigen, and we will start the development of the antibodies as soon as possible and come to the market with the most appropriate antibody pair that is required by the IVD manufacturers for the development of the rapid antigen test. In this slide, we will discuss about the development of continuously improved recombinant antibody. We have to be innovative at Fapon, because if we require like to re-engineer the immunogen in response to the circulating strains, we have to continuously evaluate ourselves and come to the market with the most appropriate antibody pairs for the IVD manufacturers. And we need to have very high sensitivity. At Fapon, when we are developing our antibodies, we already assess that the developed antibody pairs, they have the high analytical sensitivities that is required for the clinical detection at the particular moment, and they are appropriate for the IVD manufacturers to go ahead with their development. In the next slide, we will discuss about the multiple lateral flow platforms that are being used globally for the development of the respiratory antigen test. At Fapon, we developed the most appropriate antibody pairs for different kinds of the rapid test that could be based on fluorescence, on your latex particles or the colloidal gold particles. And we provide the IVD manufacturers customized suggestions how they can develop the respective format of their respiratory antigen test. Now, in the next slide, we will discuss about H5N1, which is a serious global threat. In April last year, the first human case from the cows was identified, and from that moment onwards, the WHO, CDC worldwide are keeping a close eye on how the situation will evolve, and there is a potential global threat that it could emerge into a next epidemic or pandemic, either in the selected countries like US, or it could be a global scenario. So WHO, CDC, as well as the healthcare authorities worldwide, they have already activated the IVD manufacturers to start developing the diagnostic solutions for this particular scenario. So most of the first response of the community was to develop like the confirmatory test, which is the RT-PCR. And similarly, if the situation goes out of the control or goes on a global level, there will be a need for the rapid respiratory antigen test for the population screening. So currently the developments are going on a global scale, and many of the IVD manufacturers, they have already developed or are developing the respective IVD solutions. And just recently in December, California has declared the emergency for the H5N1. Fapon has developed excellent raw materials for the H5N1, and we have already checked the performance clinically as well as analytically on a wide range of samples and panels. And several of the IVD manufacturers, they are already using our raw material for the development of their diagnostic kits. And we can also provide you uncut sheets for your respective applications. So if you require a free sample or the uncut sheets, please do send us an email. We will be happy to assist you. In this particular slide, we are discussing about the H5N1 raw materials that Fapon has developed. So we can provide you the raw materials, we can provide you the uncut sheets, and we can also do a product OEM. We have the single test, we also have like the combo test of flu A and the flu B, as you can see in the picture on the left side. On the right side, you have the raw materials for the single test as well as for the combo test. We can also provide you the recombinant antigen as well as the antigen lysis buffer. And if you need for the uncut sheet for the single or the combo test, we can also provide you that. So in a nutshell, we are providing you the complete solution for the respiratory antigen test for H5N1. And this can help you in the development of the rapid test for H5N1 very, very fast. And some of the important highlights for the H5N1 as well as for the uncut sheet solutions that we are providing. As I already mentioned, we have already checked the sensitivity of our raw materials and the uncut sheets, and we have achieved very good limit of detection in the picogram level for H5N1. We have already checked the NIBSC samples, and further we have checked the cross-reactivity, and we don't see any cross-reactivity issue against most of the interfering substances or the pathogens that can interfere with these specific tests. And we are well recognized as the IVD raw material supplier for H5N1. We have already supplied it to several of the designated laboratories and institutions in North America. And many of the facilities, they have already validated and checked the performance of our raw materials and the uncut sheets using a wide range of the clinical samples that they have at their disposal. And our IVD raw materials and the uncut sheets, they have already been checked for multiple scenarios. So the IVD manufacturers as well as the several of the scientists, they have already used our raw materials and uncut sheets for different applications. If you want to know more about our raw materials and the IVD solutions for the respiratory infections, please do visit us at MedLab Dubai. We will be exhibiting at Z2 C59. I'm looking forward to meeting you. Have a nice day. Thank you.